Yeah, it's my pleasure to present my research in this meeting. And so as Trevor mentioned, so in this project titled Endophyte-Promoted Phosphorus Solubilization in Poplar, our goal was to provide evidence of certain endophytes, a select strain of endophytes, being able to solubilize phosphorus, and provide it to the plant. And I'm hoping to convince you that we succeeded. So this was a relatively large collaboration between university and the national lab groups. And so why do we care about endophytes and phosphorus? So phosphorus is one of the critical plant nutrients, but it is largely unavailable to plants because it's bound in different metal complexes in soil. And it has to be in the autophosphate anion form to phosphate form for the plants to be able to take it up. And these bacteria called endophytes have been long known to be able to help the plants solubilize phosphates. But how they interact with plants, and in particular with Poplar, has been understudied. And so in this project, using a suite of tools, including X-ray tomography, synchrotron-based X-ray microfluorescence, and X-ray absorption spectroscopy, as well as proteomics, we tried to visualize this endophytic phosphorus uptake and understand the phosphorus chemistry during this solubilization. So we created two groups of samples. One, the control group, didn't have these endophytic bacteria, and the PMX group, which was inoculated with the endophytes. And so both groups were grown under the same conditions. They received the same amount of nutrient mix, and to both groups, same amount of insoluble tricalcium phosphate was added so that we can track the solubilization activity. So after a certain growth period, all the plants were harvested, and first we did physical measurements of root mass, leaf weight numbers, and although we did see the greatest average root length and the longest root and the greatest average leaf weight in the PMX samples that contained the endophytes, this result was not statistically significant to prove our point. Then, so first we had to show that these selected endophyte strains labeled WP5 and WP42 are indeed able to solubilize phosphate. And so this was proven by doing a colorimetric assay where these endophytes were incubated in a medium containing different phosphates. And as you can see on the figure A, relative to the control, both strains were able to solubilize tricalcium phosphate, which was critical for our study. And we also checked for two other common metal phosphates, aluminum phosphate and iron phosphate, to see the solubilization. And we found that both strains had a limited ability to solubilize aluminum phosphate. And one of the strains, WP5, had some ability to solubilize iron phosphate relative to the control. Next, we proved that these bacteria are able to colonize on the root surface. And this was achieved by fluorescence microscopy, where both endophyte strains were tagged with one WP42 with green fluorescent protein and WP5 with purple fluorescent protein. And on the left-hand side, I show a small section of a root surface where the endophyte bacteria form a likely biofilm. So we can see that these microbial populations are available on the root surface to chemically or modify the external soil chemistry. And then on the right side, where we show a section of the root with a junction with the lateral root, where we look beyond the epidermis, we can see that the endophytes are present inside the root tissue, showing that they are available for the manipulation of the internal chemistry as well. So first, some of the root sections were harvested and taken to for CT measurements. These were done both at the EMSL scanner and some at the Berkeley synchrotron micro CT beamline. What you're seeing here are segmented only root images, and from these 3D data we can calculate the accurate root volume and root surface area. So for many samples did this, and for these two pairs that I'm showing here, it was true in general that the PMIX samples containing the endophytes had a greater root volume and smaller root surface area, so indicating a larger mass growth. But the control samples had a much greater surface area, indicating a lot of fine root growth, root hair growth, as if they were trying to find nutrients more desperately. We also collected or performed synchrotron microfluorescence imaging and at the Berkeley synchrotron, beamline 1032. So these measurements, when done around the phosphorus K edge, in this case 100 eV above the edge, can provide elemental maps of the phosphorus and some of the metals critical in soil, such as calcium, potassium, aluminum, etc. When these are done on thin sections that we created, both longitudinal and cross thin sections of the roots, then we can get an insight, the chemistry inside the root, not just on the surface. So these are example images where you can see that how the phosphorus distribution and the other metals that absorb around that energy can be seen. These are some examples of, one example of the PMIX cross-sectional sample, which contain the endophytes. And here I'm showing phosphorus, calcium, and potassium distribution. So you can see that when you look at the phosphorus and calcium, in this case, some of the hotspots that are present correspond to one another, probably indicating that those are calcium phosphate present. And so these maps showed some hotspots of phosphorus hotspots, and we were able to collect X-ray absorption spectroscopy data, XANES spectrums from these spots that I'm indicating with the red. And here I'd like to summarize the results of the Micro-XRF mapping. So in general, Micro-XRF showed an evidence of phosphorus uptake in a plants with the endophytes. And if you look at the bottom two pictures where the PMIX and the control samples were compared and brought to the same phosphorus intensity range so that they are comparable, you can see a marked difference between the control and the PMIX. In the control, we see a homogeneous distribution of phosphorus along this longitudinal cross-section of the root. But in the PMIX, we see a lot of hotspots. It has this phosphorus background and the hotspot in addition. So we thought that this is the homogeneous background comes from the nutrient mix that was added to both samples, and then these hotspots must come from the phosphate that was solubilized and got inside the root. Then we performed at another beamline session at the APS at Argonne, beamline 2 IDE, fluorescence imaging of a range of samples to get statistically relevant information on the phosphorus concentrations. So in these measurements on 17 different spots, we found that the phosphorus concentration was consistently greater by 20–30% in the PMIX samples than in the controls. Again, indicating that there was increased phosphorus uptake in those groups. Next, we wanted to learn about the phosphorus chemistry inside the roots, and so for that we used phosphorus KEGG X-ray absorption near-edge spectroscopy to probe the phosphorus chemistry. On this slide, I'm just showing standard spectra. So we had to collect a range of inorganic standards as well as organic standards so that we can identify those phosphate species from our sample data. And then also beyond visual comparison, these data were used for linear combination fitting to get quantitative information. So this XANES was carried out both in defocus mode when we looked at the larger scale phosphorus speciation, and also in microfocus mode where we looked at the phosphorus hotspots. And so, and then linear combination fitting was done based on the standard spectra for all samples. These are the results which I'm just gonna go here and summarize for you. So in general, the micro-XANES suggested the chemical state of the phosphorus was a mix of organic and inorganic phosphates in both sample groups, but the overall phosphate or phosphorus and the hotspots had very different compositions. Specifically, the PMIX samples had a significant amount of organic phosphate, similar to the control, but the hotspots present in them were inorganic. The organics were phytates and DNA common in soil, and the inorganic hotspots were mostly calcium phosphates, which we suggest were related to the solubilized tricalcium phosphate. And the interesting finding that these hotspots were present, so these inorganic phosphates were present and as hotspots, allowed us to hypothesize that the solubilized phosphate may react again with calcium and other metals in the soil, in the root, and become insoluble again, suggesting that the endophyte might be able to re-release nutrients inside the plant. We turned to proteomics to look at the proteome to look for evidence of this endophytic phosphorus solubilization, and to look for differences between the PMIX and the control groups to see if any of the proteins are upregulated in one, any changes in the phosphoproteome. So the KEGG functional category enrichment analysis was is done on different functional groups, and we're looking for the most significantly changed groups. And what we found was that we found more significantly changed proteins and more KEGG functional categories in the PMIX roots. And in general, it meant that the greater access to phosphate or phosphorus stimulated more metabolic pathways in those samples. The transporter proteins generally increased in the control, which suggests that more proteins were mobilized to look for or process nutrients. Control can be imagined as a more nutrient deficient sample. But one protein, ATPase, that plays a role in cell replication, supporting increased growth of root cells, increased in abundance in the PMIX roots. In contrast to the PMIX, in the control samples, we found an abundance of a protein involved in phosphorus sensing responses, and also we saw an enrichment of oxidative phosphorylation, photosynthesis antenna protein, and proteins involved in oxidation stress in the control. In general, hinting at the more nutrient deficient conditions in the control samples. Finally, we performed string network analysis on the 100 significantly changed proteins from the previous analysis. 57 of those are shown here. This analysis works to identify known or predicted protein-protein associations based on a database. And centrality is the proxy for importance here. You can see that here, this protein called uridine monophosphate synthase, UMPS, displayed the highest in-betweenness centrality. This is a protein that catalyzes the formation of uridine monophosphate, which is a building block of RNA and pyrimidine synthesis, but it also acts as a negative regulator when there is an increased availability of sugar in the roots. It is known that when there's phosphorus deficiency in the plant, sugar from the shoots are being transported to the root as nutrients, and the disruption of this protein, UMPS, may cause an interruption in the sugar metabolism related to the phosphorus availability. So again, this provides proof that when the phosphorus availability is lower in the control roots, and we see more sugar transported there, then this protein has great importance. To summarize, I hope I show you that the Poplar samples with the endophytic bacteria exhibited a larger growth. We saw it in the root architecture, and by the control samples exhibited a increased fine root growth. We were able to visualize the phosphorus inside the root by synchrotron micro-XRF, and so that indicated that the endophytes had the capacity to solubilize phosphate. XANES studies showed that the hotspots that were identified in the PMX samples were mostly inorganic, calcium phosphate type, and this implies that the endophytes have an advantage over rhizobacteria in that they may be able to release and re-release nutrients inside the plant. And proteomics provided further evidence of this endophytic phosphorus solubilization. Finally, I'd like to thank the team who helped with this project. All the people listed here from different universities and national labs are our co-authors on the paper that has just been accepted on this project. I thank all of them, and also thank Peggy O'Day at Stanford for providing some of the phosphorus reference library to Morgan, who helped with the XANES fitting. And I'd like to thank for funding from the EMSL Strategic Science Area, Extracellular SSA in general. We are for funding for EMSL, as well as these DOE contracts for the synchrotron beamline operations. And thank you for your attention.